Hello and welcome to another Mr. Carter Science Special. In this video we'll be looking at how to carry out the starch test to find out if a leaf has been photosynthesizing. So how do we know that a plant has been photosynthesizing? Well, plants will produce glucose during photosynthesis, but plant cells cannot store glucose because glucose is soluble. So instead the plant cell needs to join the glucose molecules together to form starch, because starch is insoluble and so therefore a good molecule for storage. And you can see that in these little diagrams here. We have our small glucose molecules, and if we form these four bonds here, we can form one single starch molecule. So if a plant is photosynthesizing, it is making glucose and therefore starch. And if we test a leaf and it has starch, it must have been photosynthesizing. Yes, we could test the leaf for glucose, but that test is a little bit more fiddly and harder to carry out. So instead we carry out the starch test. And how do we carry out a starch test? Well, the first thing to do is to remove the leaf from the plant and place the leaf in very hot boiling water for approximately 30 seconds. And this will soften the leaf and kill the cytoplasm, as you can see here. Next, we need to place the leaf in a test tube with warm alcohol for approximately 5 minutes. In order to do this, we need to warm the alcohol in a hot water bath. We need to turn off all flames because alcohol is flammable, so we will not warm the water with a Bunsen burner flame. The alcohol will remove the chlorophyll because chlorophyll is soluble in alcohol, so the alcohol will begin to turn green and the leaf will begin to turn white. We can then remove the leaf from the test tube and wash the leaf in hot water for 10 seconds to soften the leaf again, because the leaf will be very brittle having been inside in the alcohol. So wash the leaf in hot water for 10 seconds and soften the leaf again. We then place the leaf on a tile and we add a few drops of iodine. Yellow iodine will turn blue-black if starch is present. And we can see that here. Here we have a leaf covered in yellow iodine which has remained yellow-brown in colour. And then here we have a leaf which has starch, so the yellow iodine has turned blue-black. This leaf here must have been photosynthesizing, and this leaf here has not been photosynthesizing. If starch is present, yellow iodine will turn blue-black. So it's important to remind ourselves at this point of the word equation for photosynthesis. Carbon dioxide plus water will turn into glucose plus oxygen if both light and chlorophyll is present. And we can use the starch test to prove various aspects of this equation. For example, we could carry out this classic experiment here, where you begin with a plant which has been destarched, so we are absolutely certain there is no starch in the leaf to begin with, and you attach a small strip of black paper across the leaf like so. So obviously where the black paper is, there will be no light in that section. And then in the other two areas, there will be some light. You then take this plant, which has, as I've said, been destarched, and you place it out in the sunlight for approximately 24 to 48 hours. Then you remove the leaf and carry out the starch test on it. And you will find that the areas which had the light will have turned blue-black, showing that they have starch and therefore photosynthesis has been carried out. The area which had no light because it was covered by the black paper, this section here, that area, the iodine remains brown, yellow-brown in colour, and therefore that area has no starch and has not been carrying out photosynthesis. So the conclusion we can say is that areas with no light have no starch. Areas with light have starch, and therefore light is needed for photosynthesis. And that explains why we place light within the photosynthesis equation. We could carry out a similar investigation here with a plant which is destarched, but it has variegated leaves. Now that means that the leaves have areas with chlorophyll here and areas with no chlorophyll, which would obviously be white. And again, we take a de-starched plant, we then place it out in the sunlight for approximately 48 hours. Then we bring it back in, we remove a leaf, and we carry out the starch test. And we have the same issue here, but we get blue-black areas where we had the chlorophyll, and the iodine has turned blue-black, proving there is starch there. And then we get brown, yellowy-brown areas in the areas where we didn't have chlorophyll. So there was no starch and no photosynthesis. So what's our conclusion? Well, areas with no chlorophyll have no starch. Areas with chlorophyll have starch. Therefore, chlorophyll is needed for photosynthesis, which again explains why we include chlorophyll within the photosynthesis equation. And our final little investigation we could carry out using the starch test would be to have two plants again which have been de-starched and set out as so. They've been covered with a transparent plastic bag, and in this plant here we have added some sodium hydrogen carbonate to make certain that we have a source of carbon dioxide. So this particular plant here has lots and lots of CO2. The second plant that we're going to compare to again has been de-starched, but in this case we've given it some soda lime, which is going to absorb carbon dioxide from the air. So there'll be no carbon dioxide within the transparent plastic bag and the atmosphere of this particular plant. Again, we take these two plants, we put them out in the sunlight for 48 hours. Then we remove a leaf from each one and we test them. The plant which has carbon dioxide, the iodine has turned blue-black, so that plant has got starch, proving that photosynthesis has been occurring. The plant that had no carbon dioxide, the leaf stayed yellow-brown, the iodine stayed yellow-brown, so there was no starch present, which means there was no photosynthesis. So our conclusion is that leaves with no carbon dioxide have no starch, and leaves with carbon dioxide have starch. Therefore, carbon dioxide is needed for photosynthesis. So those three quick and simple classic experiments prove that we need to include light and chlorophyll and carbon dioxide within the word equation for photosynthesis. Light is not a chemical, so it goes above the arrow, and chlorophyll, although it is a chemical, it isn't changed by the process of photosynthesis. It's merely there to absorb the light energy and to host the enzymes that control photosynthesis. Therefore, we just put that below the arrow as opposed to within the actual chemical equation itself. I hope you've enjoyed this short video on how to carry out the starch test in plants. If you have, comment below, let me know what you've learnt, and don't forget to like and subscribe if you've enjoyed these videos from my channel. Thank you very much for your time.